change of participants. So I'd like to presenting some of their more recent research results. And if anyone's interested after, I think there's a happy hour, and Dee said at least one drink will be sponsored. So if you want a drink, you're welcome to come out. Well, thanks for coming. So my name is David. I'm in Biochemistry Lab, and I'm going to talk to you today about a small piece of one of my research projects that I'm doing. So I studied these guys. These are the Sorry to the people on Zoom. Now you can hear me. I studied these guys. These are the red-sided garter snakes. And I'm primarily interested in many aspects of their thermal biology. So some background on them. So they live, the population we study lives in a place called the Interlake region in central Manitoba. I don't know if any of you have ever been to Manitoba, but it gets really, really cold in the winter, like -40 cold. And so immediately, these are ectotherms, right? It becomes interesting as to how they're dealing with really cold temperatures and other kind of different thermal conditions they might experience in this region. So because of the uniqueness of where they live, they have these really distinct annual phases that they go through every year. And it's broken down into these three major ones that you see here. And so we're going to focus mostly on the spring mating season. But to kind of give you the breakdown of what they are, the spring mating season is occurring right now in Manitoba. So they're doing their thing as we speak. It lasts about a month, and they do nothing but courtship behavior. And then they migrate to their summer feeding grounds, where they spend about 3 months doing all of their eating for the entire year. Then once they're done with that, they migrate back to their den sites and they go underground, and they basically just survive an 8-month winter where their body temperatures sit just above freezing for that entire time period, where it's a really stable, cold temperature. So it brings up just a whole bunch of questions or things that are interesting associated with this thermal landscape that they live in and kind of the different extreme things that they're dealing with. I'm not going to talk a lot about the other ones. We're really going to focus on the spring stuff for this talk to keep it kind of narrow. But one of the big things is, as I said, they spend 8 months kind of at a stable, really cold temperature. When spring happens, they come out, and not only are they experiencing rapid fluctuations in the environmental temperatures, average day can go from -12 to 24 degrees just easily within the day. They're seeing even bigger fluctuations in their body temperatures, where I personally was able to record animals go from about 1C internal body temperature to in the low 30s really, really quickly. So they have a really rapid kind of dynamic thermal landscape that they live in. And being ectotherms, how they deal with this is almost exclusively through behavioral thermal regulation. It gets too hot, they go find someplace cold. It gets too cold, they go out in the sun and they warm up. And they're really good at this. And this is really how they survive these conditions. Another little wrench or thing that makes it interesting in this is that this region that they live in has been really heavily impacted by climate change. So in the last 60 years, they've experienced 3 times the average warming compared to the global average. So it's kind of a big thing happening there. And primarily it's affecting the spring and summer seasons. So it's really a lot's going on right here. Where they already have a pretty dynamic thermal landscape, it's potentially getting more extreme and more changes are happening. So that's really where I wanted to focus this. So I have a whole bunch of things and avenues of research that I'm working on. Again, we're going to focus on this question here. One of the things I'm looking at during the spring mating season, which is that transcriptional response to acute thermal stress. So we talked about how they usually use behavior to deal with these temperature fluctuations. Well, my question is, as things get more extreme and things change more rapidly, there's going to be situations where maybe behavior isn't enough to deal with it. And so my question is, how do these animals currently respond to acute thermal stress events when behavior isn't enough to deal with it? So in order to look at that, I had to come up with a few ways to frame it. So I wanted to look at a transcriptional response to this. I think that that's a good way to look at how their bodies are reacting to these conditions, not just their behavior, right? I wanted to define this as acute thermal stress events, so things that happen fast, not these long-term acclimation periods, anything like that. We're looking at big swings and quick changes in temperature. And then in order to accurately achieve this, I needed to make sure that I could preclude behavioral thermal regulation and make sure they weren't able to do that, so we could really just get that transcriptional response. So we have to talk real quick about results from different research that I did, some experiments that set up, just to give you a framework for it. So real quick on this, these are some different temperature points from them, either developed gained experimentally or observed in the field. Blue is going to be real cold, green is going to be kind of normal active temperatures, orange is going to be a behavioral threshold, and red is a physiological threshold. So we're really just worried about these 3 that inform this experiment that we're working on. And so we had the CT min here, which was about 1C. This is the point when their body temperature reaches that, they can no longer escape the conditions. They lose coordination and they're kind of stuck. And so they will die if they're in that, if their body temperature reaches those conditions and it doesn't get warmer. Flip side of that is this CT max over here on the right, about 40 C, same deal. Their body temperature gets to about 40 degrees Celsius and they essentially pass out. They cannot get out of those conditions. They will die if they stay in those conditions. This one, the orange one, is a little more interesting. So this is the voluntary maximum. So this was something I was able to establish through experiment where once their body temperature reaches this point, they become in a stressed state and try and leave those conditions. And it's a very active stress state. They do not like being at that temperature or higher. And so to get a good stress response, it needs to be at least that voluntary maximum, like right there would be a good place where it starts and under that CT max. So to get this response, I defined what I considered to be an acute thermal stress event. So these are 1 hour exposures at this temperature, so sustained, where they are not allowed to behaviorally thermoregulate. So we set up an environment where they can't do that. I was able to create a couple thermal environmental chambers where I could create a really, really stable environment, like less than a degree difference in temperature through the duration of the experiment. If you see, this kind of represents that other figure we looked at, where you've got kind of the range of experiences that go from lethal to lethal. For the heat stress, we started them at the lower end of kind of normal non-stressed temperatures and raised it right up to that voluntary maximum and then kept them there for an hour before taking tissues. For the cold treatment, we started them kind of in the middle ground of their normal thermal behavior so we could get a good drop without having any kind of potential heat stress starting out, and dropped them to 2 degrees, which is just above their critical thermal minimum, right? Held them there for an hour and then took tissues. The green arrows on the side, those represent the control conditions, which were kept right about 20 degrees when we took those tissues. So I collected 4 different tissues from, we did 3 snakes per treatment. I collected brain, liver, heart, and testis tissue. I did RNA extractions, created cDNA libraries, and had it sequenced for RNA sequencing, and then mapped those results to a custom reference transcriptome for this population that we've made in our lab. I then did differential expression analysis with this. And the way that I set it up is I wanted to look at pairwise comparisons. So I went and compared the control, like for brain, the control to the cold treatment, and then a different comparison of the control to the heat treatment. Did that for each tissue, which resulted in 8 total analyses with 35 samples included in that. So I could really capture the differences in what was going on there. Okay, so I got a strong stress response or transcriptional response from all of them, across the tissues and all of it, which was great. We were able to, even in that really short period of time, get an acute stress response during this. Now there were 8 analyses. I'm not going to be able to talk about each one individually. So for the rest of this, we'll talk about them all kind of as an over unit and kind of the summary, high level summary of what's going on. So you see that there were 266 unique genes that were significantly differentially expressed across tissues and treatments. There's obviously overlap between each of those categories, but there were 266 unique ones. And they were split 50-50, that there were 133 unique up and 133 unique downregulated. What I think is really interesting is zeroing in on those genes that were commonly differentially expressed to see like which ones are really important for these conditions that they're facing. And if we look, so to orient you really quick, the numbers represent number of differentially expressed genes. Red numbers are upregulated and blue numbers are downregulated. And if we look at the acute heat stress, we see that one jumps out immediately as being something that could be interesting. And it's this one. There was a single gene that was significantly upregulated in all tissues for that treatment. So I wanted to dig in a little more on that. So what it is it's a heat shock cognate 71 protein, the HSPA8, and it's just a member of the heat shock protein 70 family. It has its main function, like most of them, involved in protein like chaperone, helping refold misfolded proteins, and this one specifically plays a big role in just kind of normal maintenance of helping fold newly synthesized proteins. It's constitutively expressed, so it should always be on, kind of just working and doing general functional maintenance on the proteins. But it has been shown to be inducible under heat stress, thinking that this is likely an early stress response, that this plays a role early on, helping with it, and then other things kick in later. We do see that it was not just upregulated in all heat treatments per tissue, but also half of the cold treatments, which I found to be interesting as well. But I was curious about the 2 that didn't have it upregulated. and found that there was actually a different heat shock protein that was upregulated in those ones only and wasn't upregulated anywhere else. And so this is the HSPH1. It also is just, it's a heat shock protein. It does a lot of the normal stuff, but it has an additional function where it prevents the aggregation of proteins in cells under severe stress, right? So this may be more of a later response, potentially, as to what's happening once things start backing up and the function's not working as well. Additionally, though, it's been shown that it inhibits the HSPA8 activity. So it would also make sense that those wouldn't both be upregulated at the same time because they're kind of having some effects with each other. Of course, oh, you can't see an important part. There were a ton of other heat shock proteins upregulated. This is expected. They all work together in different ways, and there's kind of a robust response. But one that was notable is one you can't see, this HSPA14. It was the only one downregulated. It was only present significantly in testis, and it was downregulated in both, and relatively by a lot. So this was really notable. I looked into it, and I couldn't find a reason based on its function why this would make any sense. However, I did find some other papers that were published that found the same thing, and their conclusion also was they had no idea as to why it would be significantly downregulated. So it's kind of an interesting one that's worth looking at further into the future. Okay. So we have the same thing here. On the acute cold stress, there was a single gene that was significantly upregulated across tissues, right? So again, this one is probably pretty important to that process. I wanted to look into that. So what we see here, so this is the synaptogenin 2. It's a phosphatase. It's associated with a whole bunch of different things, much of which is around the membrane. It does a lot with vesicle transport, the clathrin-coated vesicles. And currently it's in a ton of cancer research papers right now. It's something they're looking at for that. But one thing that I found interesting is that it is also known to be an effector for the RAC1 gene, which is a RAC family GTPase. So if it's, you know, potentially playing a role with another gene, I wanted to see what does that gene do? And the answer is that gene does everything. It is involved in tons and tons of different cascades and signaling pathways and all these things. But one of the things that it does is it is involved in heat shock response. So I was like, sweet, this is a good candidate. So I looked and I'm like, okay, it's not in my differentially expressed genes. Turns out it's not in my non-differentially expressed genes. It wasn't mapped to my reference transcriptome at all. This is a ubiquitous gene that should be everywhere, and it didn't show up in my data, period. I checked my reference transcriptome, it is there. So something's going on. I lost it during the mapping phase. So I'm gonna have to go back and look and see if I can find this. But it was interesting how that path took me from one gene to another gene to like, where is this gene that really should be there? But yeah, so we're getting close to the end of time. So basically, the kind of conclusions from this little bit here, I was able to get a strong response to this, even with this really short time period. So it does show that when they can't behaviorally thermoregulate, they do have a fast acting mechanism to help them deal with that and survive those conditions. I don't show all the data here, but with some functional enrichment stuff, it also showed that the response is pretty similar between heat and cold stress. They're using a lot of the same things, turning the same things on or the same things off. And then of course, I found a couple potential key players that warrant some kind of further investigations to see exactly why they're so good in that. Obviously, additionally, I'd like to look into finding that RAC1 gene and do some digging in my early data, and then looking more into why that other heat shock protein might be downregulated. Also, just a couple other quick notes. So the range for TSP is this yellow portion here. It's huge. And we're just looking at them, I can't even reach it right here, kind of on the more northern side. And I think that a lot of the thermal stuff I'm looking at would be really interesting to look at across their range, specifically, you know, down into northern Texas and things, and see kind of at, you know, behavior, physiology, transcriptional, and even like evolutionary level, how they might differ just based on latitudinal difference. I think it would be really, really interesting to look into that. Oh, and additionally, this was all, everything I talked about today was just in spring. It's all physiology and whatever during this spring month. I think that it could be very different in summer and fall, also in winter. So it could be really interesting to repeat some of this in these different seasonal time points and see how they might be different. Okay, that's what I've got. Thank you.
We have a question from Jimmy Cornelius.
Okay.
Hi, can you guys hear me?
Yes.
Awesome. That was really interesting talk, David. Thank you. I was thinking about that heat shock protein in the testes being downregulated. And it struck me that the testis is one of the like classic trade-off tissues with survival, reproduction versus survival.
Mhm.
And I mean, I don't know how limiting any of these protein components might be, but it just struck me as interesting that the testis was doing something different than all the other organs.
Yeah, and actually I have another slide. It turns out there's a lot. It has a lot more of the downregulation happening within the testis. Not all of it's shown here, but there's a lot of things being turned off. Everything involving reproduction is pretty much shut down in them in both the heat and cold stress conditions. So yeah, it's a really good point, and there's definitely a lot of activity going on within that specific tissue in response to temperature. Yeah.
Yeah, thanks. That's a really nice talk, Dave. I wonder if you have plans already or are thinking about DNA sequencing as well for these regions, like to follow up if we need to look at, especially for these heat shock protein genes, the variation within them, particularly along with like a latitudinal gradient.
Yeah, I would love to do that. I am doing some DNA work. We can pop back to, I can never get to it. I am doing some DNA work here where I am looking at recent genomic change, potentially in response to climate change. I'm hoping I have historical samples from 25-ish years ago that I've compared to modern samples. I'm not done with the data analysis on that, but that wasn't a targeted one. It was more shotgun. So I'm hoping to find some stuff in there, but yeah, I think it would be really interesting to do more of a targeted approach, looking at like maybe some of these specific heat shock genes in different regions and in potentially the old population versus the new population. I think that's a really good line of research. Yeah. Yeah.
There's another question. I was wondering if you will be looking at how the snakes may respond to dramatic acute shift, like the spring we are seeing now, where yesterday is warm and today is pouring. How will the snakes handle that?
That is part of what I was trying to capture here, right? Is that they come out, so I'm not sure if many of you are familiar with heat hardening, but continued exposure to high temperatures can change their expression levels for genes associated with that. And I tried to capture before that happens. So when they're coming out of this really cold, long, stable time period, and they're moving into this really dynamically changing environment, that was the whole point of why I wanted to look at this acute stress response and not more of a long-term acclimated one, but this like really quick changes in temperature. Okay. Yeah.
Yeah, it's kind of more from a technical kind of thing. The rack one story is interesting. Just to confirm, did you say nothing mapped to it?
Yeah, nothing. I did not find anything that mapped to it. Yeah.
Any treatment, nothing. So,
no.
So given you did TAG-seq, there's a chance, right, that your reference may not have the 3' end well covered.
Mm-hmm.
TAG-seq covers poly tail a little bit into the gene.
Yep.
So you might not have overlap in the reads once you reference it.
Yep. That was one of the things I was going to look for. Also, maybe a potential partial read where it would have double mapped and just thrown it out during reading. So I haven't had a chance to look for that yet, but those are things that I'm definitely planning on looking at, trying to figure out what's going on there. Yeah.
Seems like your data might be really well suited for gene co-expression network analysis. So you have all these, kind of similar experimental designs in different tissues.
Mm-hmm.
Have you thought about doing that? Basically, kind of boils down to correlation coefficients.
Yeah, I'm not sure on the exact phrasing on that, but I did do gene set enrichment analysis on it, which I think is slightly different. Yeah, okay, sorry, I'm not super familiar with that one. So the answer would be no. Then I haven't done that on it at all.
I mean, it's not too tough analytically to do, and it kind of shows you sets of coordinated differentially expressed genes.
Okay.
So you might be able to sort of say there might be a few major events.
Awesome. All right.
Anyway.
Great. No, I will look into that. That's awesome. Thank you. All right. Cool. Well, thank you.
Is this on? Yeah, okay, great. Hello, folks. So I'm Bryan Lynn. I am fourth year PhD candidate, advised by Patrick Deline here, and then also Martin Schuster over in Micro. And my project looks at using the CHEMOSTAT to examine fundamental questions surrounding the evolution of cooperation and the tragedy of the commons. And admittedly, there's a lot to unpack in that sentence. So I want to start with why we might be interested in looking at cooperation. So I'm going to define cooperation as an act that has a cost to the individual, but then benefits the recipient. So if I was a squirrel and I shared my food with another squirrel, I now have less food to grow and survive on, but the other squirrel is very happy. It's all over nature. It's across different levels of organismal complexity, from humans to, as you'll see, spoiler alert, also bacteria. But it's really hard to explain through like our classic Darwinian survival of the fittest lens. In fact, Darwin even noted it as being contrary to his theory of natural selection, but being the boss that he is, he just ignored the thing that contradicted him and persevered. That is all just to say that natural selection then would favor the selfish behavior because it increases the fitness of the individual to not share their food as a little squirrel. This favoring of selfish behaviors can result in the invasion of non-cooperative individuals that we refer to as social cheaters. And then that invasion of these non-cooperative individuals can also result in a population collapse or resource collapsing. This is a phenomenon known as a tragedy of the commons. And so the evolution and stability of cooperation is an enduring problem for many social dilemmas and social systems, such as responsible use of shared resources. So I am trying to contribute to our understanding of how cooperation, or the lack thereof, is going to impact population dynamics. And I'm specifically doing this by investigating the socially cooperative bacteria Pseudomonas aeruginosa in a chemostat environment, which leads to the question, how the heck does bacteria cooperate? So bacteria such as Pseudomonas aeruginosa exhibit a variety of behaviors, including biofilm formation, microbial warfare, and collective nutrient acquisition. But what I focus on is how Pseudomonas aeruginosa produces an extracellular enzyme, which is protease. And that serves as a public good, as it's extracellular, it's just kind of in their environment, and it's shared by the entire population of cells. So more specifically, how this works is that the bacteria is going to produce a signal molecule, and as the population grows, so too does the quantity of that signal molecule that's in their environment. And at a certain threshold, there's enough signal around, they're going to be able to start receiving it. And once they receive this signal molecule, it's going to turn on, this is a LOSR gene, and turn on all the things that that gene expresses, including the production of this protease enzyme. So once the population of cells hits this threshold and starts producing the enzyme, they're a happy population and they all eat this, they all benefit from the enzyme being in their environment. The role of the enzyme is that it breaks down larger protein sources into smaller bite-sized ones. So for humans, this might feel like the difference between being in a field of cows and being at a steak restaurant. Under one circumstance, you can eat and grow and digest, and on the other ones, you're just kind of vaguely aware of being surrounded by beef. However, we can knock out this gene, and these are social cheaters who can then not produce this enzyme. Now, not producing the enzyme means they're not spending energy producing the enzyme. So they have a growth advantage, and they do in fact grow faster. And that growing faster poses a burden on the population as a whole, because as they begin to outcompete the cooperating enzyme producing bacteria, we get less and less of the enzyme in the environment. And when there's less enzyme in the environment, there ends up being less food to eat, and that results in our tragedy of the commons. Okay, so why the CHEMOSTAT then? Why I chose this place to this study system? The CHEMOSTAT actually is quite similar to larger ecosystems. So to draw an analog between a population of vampire bats. In both systems, we have nutrient entering the system. With the bats, they go bring a blood meal, bring that back into the cave. With the chemostat, simple, I have a pump that I set it up to that's in red. Of course, we also have the population turnover being expressed in blue. Bats are going to leave through migration or death. On the chemostat, again, I just have a pump. It's a little bit simpler. And then we have the cooperation and dynamics between the population. So the vampire bats are going to share their blood meals with one another, whereas my bacteria does that whole enzyme thing I just talked about. So the chemostat's a really ideal environment to look at some of these things. And so then it's kind of like, besides just being fundamentally super interesting in my unbiased opinion, one reason to care is that bacterial antibiotic resistance is continuously posing a challenge. And it might be possible to use the social behavior of microbes to actually treat that. So my goals are to use the chemostat to investigate these questions, looking at whether the bacterial dynamics match theoretical predictions. I'm also using some of that theoretical framework to explore ways we might be able to sustain cooperation. And then I'm doing a rigorous marriage of the theoretical and empirical work. This is obviously a short talk, so I'm not going to do all of those today. I think just 1 in 3. And then how I'm doing that, the math modeling, the chemostat operation, and then the within that, we also are looking at what percentage of the population are cheaters, and we can do that genotypically and phenotypically. And I'll get into the details of how that works in a second. I am going to start with talking about my model, and I know that like the vibe is usually that we don't love quantitative stuff, except for Ricardo and me, but I'm going to do it anyway. So this is my very scary model, but what we're seeing modeled here is the substrate being pumped in at a dilution rate and everything being pumped out at the same rate. We see the substrate enzyme turnover. So this is the enzyme meeting up with the substrate and turning into the steak, if you will, digestible products. We have the uptake of those products by the bacteria. We have the enzyme being produced by the cooperator, and then the reduction of growth due to that production that the cooperators experience. And then we have the growth of the bacteria as well. Of course, I have the chemostat that I'm running. And then when it comes to determining the percent of cheaters in the system, our gene knocked out cheater is also antibiotic resistant. So I can grow them on a standard plate and an antibiotic plate to get a total population size and the population of my loss-R cheater. We also have the protease deficiency, which is more of a phenotype look at the system. So the enzyme will actually, on these white milk plates, clear out the white color. And so I can look at whether there's enzyme production based on the clearing of that protein, take a diffraction of how many are doing the phenotype. So this is the exciting part, the results. When I grow just the cooperators alone in the chemostat, we see a stable population over many hours of growth. And by the end of it, we do see a small number of cheaters evolving, which is expected when you grow bacteria for almost 200 hours. With the coculture, which is where I have cheaters and cooperators together, we did get a population collapse. Intriguingly, we saw really divergent cheater frequency trajectories. And I'm gonna dig into some predictions about that. But first, it makes sense to talk about how I integrate that model with my empirical data. So some of my parameters are determined by experimental design. The dilution rate is set to how I set the pump rate. We had parameters that could be measured during the experiment, so I can actually quantify the growth difference experienced by my cheaters and cooperators. And then some parameters had to be estimated from other research. And after a lot of effort parameterizing my model, I got a model that I think fits like really well. I'm super happy with it. This is one of those figures that's like 2 years of work, and it's like a tragedy that it's 3 seconds on a slide. But this is my, in my mind, this is my like, ta-da! Wow, what a great model! Yeah, so it's just all bask in its glory. Okay, but so then I can actually use this model then to explore possible explanations for that divergence. So one thing we thought about are mutations. Bacteria love mutating. So there's faster growing mutants could be the cause. I looked at whether there was a faster growing cheater and adding a faster growing cooperator mutant and found the dynamics match theoretically. Another thing we looked at was that slightly variable initial conditions might impact long-term trajectories. I'm sorry for how large that figure is, but the main takeaway is that we ran lots of simulations under lots of different variable values, and some parameters definitely had just a small amount of impact, and then others had a larger impact on long-term trajectories. So that's a possibility. Stuff we haven't explored yet include biological processes. There's so many things we could think to include. One is that yeast has been shown to be able to partially privatize the public good they produce, which essentially means when they produce their enzyme, they benefit from it kind of being nearby first. Another thing is that maybe it's genetic drift playing a role, since the weird frequency changes happen when the population density is really low. So what's next? More experiments, because that's what reviewer 2 said. So can we use our CHEMOSTAT samples to support the idea that there are faster growing mutants? Stay tuned, I'll find out next week. And then I'm also incorporating spatial structure by investigating how biofilm formation could impact these population dynamics. So we treat our CHEMOSTAT with this equivalent of a Rain-X to try to keep biofilm away, but we want to see if maybe that has a role to play in the equation. So I do want to thank everyone that's given me academic, emotional, and/or financial support. And then questions if anyone has some. Yeah, Andy.
Curious about your thoughts on drift. It's a possibility. When I think bacteria, I just don't think drift. So I'm curious how whether the specific dynamics of how small the population might get during the phasing, and what it might be typically with the experiment.
Yeah. So I mean, what we're looking at, so here we have the population being pretty low. OD for one of the replicates was below detectable. The other one was around between 0.1 and 0.05. And so the thought is that because we have this dilution that's kind of going through and flushing it, that if there was a subpopulation in there that did have a different trait, that could play a role. The reason we didn't explore it or model it is kind of what you're saying. We chose to explore further the explanations that we thought were most likely. But I do want to acknowledge that that what I explored was not an exhaustive list. There's many things that could be happening. It's
Yeah, so I guess I have more of a general question. So I know in a lot of the literature on cooperation, usually they invoke kin selection and things like that. So how do you think about that in a bacterial framework, right? Presumably these are starting off from some kind of
Yeah. So we do look at what you're referring to it, what we really think about is mechanisms that sustain cooperation is the verbiage if you wanted to Google it. So how we think about that. One of my projects that I didn't have time to talk about right now is looking at policing as a mechanism. And so what does policing look like for bacteria? It's toxin production. And so we think, does the cyanide that this bacteria produce, could it be used to, if the same gene that's responsible for enzyme production is responsible for the production and being immune to the toxin, then it could be served as a punishment. Other things I don't know if kin selection is going to show up a lot in bacteria. Well, yeah, I mean, I guess you could also think about different signals and receivers as being a form of kin selection. We could have, I know that there's nutrient acquisition, siderophore which is a little bit of like microbial warfare is like another mechanism. So if you have like the matching pieces with that one. So yeah, there's surprisingly like a lot of these mechanisms will relate to the bacteria in different ways. But yeah, that is part of my project, just not this one.
So have you ever seen any evidence of something like frequency dependent selection in any of your projects? Like I'm looking at that figure and I'm wondering, like, if you went longer, you know, what would happen to like, the blue replicate in relation to the red replicate? Or, you know, I'm curious if there might be longer term dynamics that would be interesting that might play out.
Yeah, I mean, once we got this figure, there's of course play, well, what would have happened if we ran it longer? We're a little bit limited in trying to keep the chemostat to be sterile. So when we assemble it kind of all together, we just have this one like 4-liter container full of the media, the growth medium, the like food that we send through all the tubes. And of course I can't even fill that all the way, but so when we designed it, we thought 200 hours would be a long time to see dynamics, but it's totally possible that expanding that you might see recovery, or you might see, who knows, oscillations. Our hope was that 200 hours would be enough. I don't know, is it ever enough? Should I just grow them forever? How long are you guys going to want to fund me for? Yeah.
Sort of along the lines of your follow-up to Mark's question, I'm curious how like the role of sort of multi-level selection fits into this. So, and just for the record, I know you were kind of joking, but rather than just move on and ignore the problem of cooperation, Darwin actually invoked group selection, right? I would say sort of non-naive group selection. And so, yeah, how does that all fit in? And I guess is there a reason in the sort of life history of these bacteria that between group selection would be a relevant force in this case?
Yeah, so in the world of like folks that study cooperation, group selection was a hot topic for like a decade or two. I might get the decade wrong. I want to say it's 80s, but and it was like super looked into, but basically everything came down to that selection operates quicker at the individual level. And so there was no evidence to suggest that the group selection was capable of maintaining anything, because the second you have like a cooperative group, you get one cheater that wants to be like, hey, I want to infiltrate this group, and now you wipe them out. So at least with the like way it's modeled and way it's understood in the classical sense, group selection has been like a not as promising of an explanation as one would think and hope. I don't know, did that answer the
Let's
question?
talk later. Yeah, I think that's true in some cases and not true in many other cases.
Yeah, and what I think that brought into, I think that's probably what led into this fold of like being really interested in the mechanism that sustained it. Because like, what do groups have to offer then that like can keep it at bay, right? So they can offer the community structures. So again, like this is also a well-mixed solution, like populations. Like what happens when you have population structure, which is why I'm looking into biofilms next, because population structure is shown to be like very successful for some of this maintenance. But within that population structure, you might have things like the policing of behaviors and ostracism and all those other conditions that have been shown to have an effect, which is part of being in a group, but just the group itself isn't enough. Question, right? Does that answer it?
Yeah.
Okay. Yeah. All right, pass the torch time.
Hello? Yeah, okay. 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 So, hi, I'm Shumpei, and I wanted to share a story about the importance of experimental context. I wanted to share this story because I thought it'd be a little bit broadly appealing instead of the other stuff that I work on. And it's a story about the limitations of using cultured algae to study Cnidarian algal symbiosis. So first, diving into Cnidarian algal symbiosis, this is an intimate partnership between cnidarians, which are the hosts, and dinoflagellate algae, which are the symbionts. So when I say hosts, I'm referring to the Cnidarian animal. And when I say symbionts, I'm referring to the dinoflagellate algae. And in this symbiosis, it's a mutualism, so both partners benefit. The symbiont provides photosynthate to the host, so they photosynthesize, give off their sugars to the host, while the host provides protection and nutrition to the symbiont. And this symbiosis is key to the function of coral reefs. And as you can see, coral reefs are very beautiful. They're incredibly hot, or they're diversity hotspots. They're important for fisheries, they're important for tourism. And I don't think I have to convince you guys why corals are important. But here is a bleached coral. So this is a phenomenon known as coral bleaching. And this is a coral that's had its algal symbionts expelled from it. And this is largely a heat-induced response. So it's a heat-induced release of algae from the host. And corals without their algae will starve to death eventually and die if temperatures do not go down. So if temperatures do go down, they can recover. So there's potential for recovery, but if they don't go down, then they will eventually die. And you can imagine what kind of havoc that could have on a coral reef. The climate crisis is causing global bleaching events where entire swaths of corals globally will bleach. And corals are basically threatened for extinction if climate change is not mitigated. And of course, stopping climate change is the best way to save corals, but there's also a race for solutions to save corals. We're gonna science the heck out of it, right? So to fulfill that quest, understanding the basic biology of the symbiosis is critical for the development of these solutions, right? So going in a little bit more specifics about the symbiosis, and this is gonna be a very algal-centered perspective of the symbiosis. So in this symbiosis, the algal symbiont resides within host gastrodermal cells. So here we have two tissue layers up there. On top, we have the ectoderm. Right below that, we have the gastroderm. And the algae live within the host cell. So this is an intracellular symbiosis. That's what I meant by intimate symbiosis, right? So the algae are in a host cell environment. The pH is around 4. It's very acidic. It's an acidic organelle. And it's protected from the environment, right? And the algae can also be found ex hospite. So in hospite meaning within the host, ex hospite meaning outside the host. And when it's outside the host, it's in the ambient seawater environment. The pH is whatever the seawater is, 7.7 to 8. But they're independent. They're not protected, right? They're fending for themselves. So these environments are very dramatically different from one another. So the symbiont must undergo molecular changes to switch between the two lifestyles, right? I forgot to mention here, the algae can exist in stable populations, ex hospite, but they also actively released by the host. And it's in this case, it's not a stress response, it's just a homeostatic mechanism where the host expels algae regularly just to maintain a stable population of algae. So people have compared the biology of the two, of inhospite and exhospite, to better understand symbiosis. So people have looked at transcriptomes of algae, comparing inhospite versus exhospite. And the classic story, what people commonly find is differential regulation of nutrient transporters. So in this study, the most differentially regulated genes were that of nitrate transport and ammonium transport. And these were upregulated in hospite, so they were upregulated within the host. And these are classic indicators that the algae are starved of nutrients. And the reason why that is because if you're an algal cell, you have plenty of nutrients around and you're getting diffusion of the nutrients inside. You don't need these transporters to get enough nutrients, right? When you're starved of nutrients, you're trying to scavenge as much as possible, so you're overproducing these transporters to get as much as possible. So these are starved of nutrients in hospite, right?
So here's the classic understanding of symbiosis for the algae. Algae ex hospite, they have plenty of nutrients, so they can downregulate their nutrient transporters. Whereas algae in hospite, they have to upregulate their transporters because we think that the host is controlling, they're limiting the supply of nutrients to the symbionts. And if there was a textbook for Cnidarian algal symbiosis, this is what you would learn. This is what people would tell you. This is what I learned when I started this kind of research. But the problem with this is that algae ex hospite use cultured algae. And cultured algae are grown in a nutrient-rich media, so they're given a lot of nutrients, right? Whereas algae in hospite are sampled from corals that are in just normal oligotrophic seawater, in low nutrient seawater, right? So this comparison might not be reflective of symbiosis. It might just be a factor, an artifact of experiment, right? So context matters for these kinds of comparisons, right? So here we have that same model, but now we're introducing culture here. So algae in hospite, they're in oligotrophic seawater, they're in low pH, and because they have low nutrients available to them, they have low cell division rates. Within the environment, they're in oligotrophic seawater, just like in hospite, ambient pH, but we don't know much about their cell division. It's not, a lot of the biology of what's going on here is not very well known. Algae in culture, they're in high nutrient rich media. It's the pH of the culture media, and they have high division rates because the culture environment is basically optimized to benefit the algae, right? So culture clearly does not reflect what is found in nature. So what if we sampled algae that were released from the host here? Would that be a more natural representation for comparing between the two lifestyles, right? So in our lab, we use Aptasia. Aptasia is a sea anemone model for the study of symbiosis. And in Aptasia, the algae are released in discrete pellets called egesta. So here's an anemone mouth close-up, and it's a video of slowly releasing a pellet of algae. Here's what it looks like right next to the anemone, and here's a much more close-up of the egesta. And the egesta are chock full of the algae. There's not a lot of other things in it, so it's a very convenient system. You can sample it very easily. You can look for a pellet, sample it, and it's full of the expelled algae, right? But again, very little is known about the biology of the algae from egesta. And our goal was to figure out, is this a good model to represent the ex hospite state for future studies? So we looked at the basic biology of the algae from egesta, because we don't even know if they're alive. We don't know if they're digested algae. And then we also tested the expression of those nutrient transporters that were differentially regulated. So first, we looked at division rates. A reminder that culture cell division was high. Algae in hospite cell division is low. So here's mitotic index. This is a measure of cell division. And we have algae in hospite, and we have algae from egesta. And these are paired data. So this data point here is from an anemone, a whole anemone that we ground up. And then that other corresponding data point over there is that egesta that it released. So it's a one-to-one comparison. And you can see algae from egesta have a higher division rate of algae than in hospite. So this suggests maybe that there is some kind of control that the host exerts. Maybe there is some limitation of nutrients. There's also the hypothesis that the host might be preferentially expelling algae that are dividing, just because when algae divide, they get bigger. So if you just spit out the bigger ones, that might be a good way to get rid of dividing algae to maintain your population. But that's all conjecture, right? So we don't know. We don't know what's happening here. But so far, egesta might be more similar to culture than we might think. We also looked at the reestablishment of symbiosis. So you can take a symbiont-free anemone Aptasia, you can add algae to it, and you can reestablish symbiosis, you can get symbiotic Aptasia. And you can measure the symbiont density within the Aptasia to get a quantified measure of success of reestablishment. So in this experiment, we looked at cultured algae, we looked at algae from egesta, and we looked at freshly isolated algae. Freshly isolated algae is just an anemone that's been ground up, and we got rid of the host material and just kept the algae. So first, here's symbiont density, and we have culture compared to freshly isolated. You can see algae that are freshly isolated do better at reestablishing symbiosis. And this is a result that's been seen in the literature decades ago, but we still don't know why that is. It might just be that there's host material in the freshly isolated algae, and there's some kind of self-recognition going on that the host likes things that were already in it, something like that. Not sure what's going on there, but here's how egesta fits in, algae from egesta. So you can see it just does just as well as culture, but it's very different from freshly isolated, right? So egesta falls in line with cultured algae. And so the summary so far, at least with the 2 data points that we've looked at, egesta are competent symbionts, they're alive, they're metabolically active, and they can reestablish symbiosis with the hosts. And they kind of so far mirror characteristics of culture, right? So now let's look at the thing that we really want to look at, nutrient transporter gene expression. So we looked at gene expression, we tested it with RT-qPCR. We looked at 3 nutrient transporters, at least gene copies of them: ammonium transporter, nitrate transporter, purine nucleoside permease, and a nitrogen handler. This is nitrate reductase. It's involved in converting nitrates to ammonia. Ammonia is just the more preferred form of a nitrogen compound. And these are all known to be upregulated in low nutrient conditions. And all this work that you're about to see is done by STAR undergraduate Julia Unsworth. So what you're about to see is all thanks to all of her hard work. Okay, so here are the genes. We have AMT, ammonium transporter, nitrate transporter, purine nucleoside permease, and nitrate reductase. And we have in red, we have culture, in blue, we have egesta, and yellow, we have algae and hospitae. And you can see that there's no significant difference between algae and egesta and algae and hospitae for these genes, for all of them. And then algae and culture, comparatively, they're all downregulated. So a reminder, all of these genes are upregulated when you're starved of nutrients. So it seems like egesta are just as starved of nutrients. So maybe there is no host control happening, right? It's just that you're comparing it to culture, which are in a nutrient-rich environment. So here's our shift in thinking. So here's the old model. We have algae ex hospitae. They have nutrients. They don't need the nutrient transporters. Algae and hospitae, we have host control of the nutrients. So you need the nutrient transporters to try to get as much as possible. But then when you put egesta within this picture, you can see there's the nutrient transporter is still upregulated. And that's because there's no nutrients available because they're in oligotrophic seawater. So in algae and egesta, algae and hospitae are both in oligotrophic seawater, whereas algae and culture are in nutrient-rich media. So what's really happening there, it might not be that the host is exerting control over the symbiont. It's just that there's not any nutrients to begin with, right? So that's this new perspective. In conclusion, what does this all mean for corals, right? So now we kind of have a better understanding that the host may not exert as much control over nutrients to the symbiont as previously thought. And this is important because corals, when they're exposed to elevated nutrients, are more susceptible to bleaching. And so now we have a more biological, mechanistic understanding of why that might be. The host isn't really controlling nutrient availability. It's just bringing in whatever's there, right? We also want to know what other genes are differentially regulated between algae and hospitae and algae from egesta, because we want to know what makes symbiosis possible, what's differentially regulated with symbiosis and not with has nutrients being a confounding variable, right? So we want to test this. We want transcriptomes, we want proteomes of this comparison. I'm not going to do it, but hopefully someone will. Maybe I'll do it in the future. I don't know. Okay, and with that, I'd like to thank you for listening. Thank NSF for funding this. Thank Julia for that qPCR work. Val has also helped me with this project, and Virginia, my advisor, as well. Thank you. Yeah.
Just the expression stuff from the egesta algae.
Yeah.
Were they done in culture, in media, or is it done in the oligotrophic, from water? Because you have the nutrients, right? You said there's nutrients there.
So yeah, the egesta were sampled from anemones, like the same container That, so
I get it.
we didn't move them, yeah.
Yeah, I guess maybe I missed something, but I'm still convinced that the lack of difference in expression
Yeah.
of those transporters of egesta versus in-house
Yeah.
means that the host has no control. Because
Yeah.
it means that there's low nutrient availability for the algae, where one may be the host controls the nutrients, the other one there isn't any water.
Right. Oh,
there's no
so
lack of nutrients available, so the transporters go up. But
Right.
saying that the host, that means the host has no control, that's quite, it's not.
okay, yeah,
sufficient.
I can see that. So it would be better if there was another experiment where
You didn't?
you have the aptasia in like a little bit more nutrient-rich environment. And then you could try to test that.
Yeah, you did not remove the host. You know.
That's a good idea,
You
actually.
use- That's right.
Yeah, I agree with that point.
Curious about the culture treatment, kind of how long that culture's been established. You might imagine that if it's a long-term algae culture, evolutionary dynamics might come into play, like adapting to the class, relaxed selection on symbiosis stuff.
It's definitely something the field is just ignoring because the cultures are decades old and they just use the same line and they don't care about whether it's changing or not.
Mm.
Yeah, it's definitely a concern. Yeah, like the algae are just have evolved to live in culture and not care about going back in the host, right? Yeah.
Right.
So far, they do go back fine, and they are fine symbionts. Yeah, anemones are happy, but yeah. I like your comment, Felipe. There are studies, if you go back to the literature, there's a good amount of evidence that when you add nutrients to the seawater, that the algae just take it up. There's no control of that. So the effects that you see from algae in culture when you add up nutrients or when you take it away, you see the same thing if you put it in the seawater with the host. But just in this vacuum, yeah, you're right. Yeah. Do we have any comments in the chat? No. Okay. Okay, thank you. Should we stop this?
You said you'll cover drinks for us, at least one of them. Drinks and snacks.